Are you guys ready with that camera?
Yep.
Okay, so if I can get everybody's attention, I'll introduce Gemma. Gemma Bales comes from UCL, so University College London, and she's visiting us. This is actually her second visit. She was here last summer, and she's here for 2 months, and we finally squeezed in a talk. She said she's going to be here for, I think, 3 more days. And I think Gemma, being in Canada for so long, is now an official Canadian, because at the end of her last trip, she was actually at the last concert that tragically hit with Prime Minister Trudeau. How much more Canadian can you get? So anyway, she and her PhD supervisor, his name is on there, Passivity, Dr. Passivity, they do a lot of work in broadband, and we've been had lots of discussions with them over the years at conferences and followed their work, particularly with cytochrome space, which I'm going to talk about. So I'm going to turn over the floor to Gemma right now, and then everybody interrupt to talking about, I guess, 5 or 10 minutes, and then we'll bring you to something that shows up. Okay, so.
Thank you very much, Steve. Thank you for inviting us to talk today, and also for allowing us to come and collaborate with your lab. So I'll give you a quick background about me. So I'm from the UK. I grew up in Shropshire, which is in the middle of nowhere in the Midlands of England. The only notable thing about us, apart from the amount of hills, is that Charles Darwin was born there. So I thought I would introduce my background through Charles Darwin and sort of align myself with him for you. So yeah, I grew up in Shropshire, but then I moved to London to do my undergraduate degree. I studied physics. Unfortunately, Imperial College London started 6 years after Darwin died, so I don't have a link there, but bear with me. I then went to Cambridge, where can everyone hear me? I went to Cambridge, where I did my master's in optics, like Darwin, who also studied in Cambridge. And for the last 6 years, I've been at UCL, which is in the centre of London, and I did my PhD there in biomedical optics. And also the building that I work in now is where Darwin lived while he was in London. So now I've introduced myself and sort of aligned myself with Darwin, I can tell you why I'm here. So I come from London to London, and that's caused a lot of confusion in talking about where I am and what I'm doing. So I've been trying to name this London fake London, although people here seem to have a bit of a problem with that. So as it's a research talk, I thought I would provide some evidence. So although London, Ontario does have an earlier evidence of settlement, London, England was named London first, so the Romans named it Londinium in the 2nd century. So there's a bit of a debate there, which is the first London, but we do have a bigger population than you, so I think we would win the battle. So for the purposes of this talk, I will be referring to this London as fake London and London, UK as real London. We can discuss in the questions afterwards if you like. So onto the science. So I'm going to give you an overview of what I've been doing. So I work with near-infrared spectroscopy, which is a biomedical optics technique. And in particular, I measure cytochrome C oxidase, which is a metabolic marker. So I'll talk a bit about that and how we measure it. And then I'll present the work we've been doing in the neonatal intensive care unit in University College London Hospital, looking at primarily babies that have had hypoxic ischemic injury. I don't know what's happened. Pizza time. Yeah, so we'll enjoy pizza and then I'll carry on. Or fix this. I don't know, the screen just went. and some of our analysis of those studies. We're looking at both hypoxic, ischemic encephalopathy, which is a brain injury in newborn babies, and we're also looking at babies that have abnormal brain function, such as seizures. And then finally, I'm going to present some of the work that I've been doing here in Fake London in collaboration with Keith's lab. So this is a little movie that we've made just to give you an overview of the measurements we're making. So here we have a neonatal brain, and if we zoom in, we'll get to the neurons. And in those neurons, there are lots of mitochondria, which are the powerhouse of the cell. And every time that the mitochondria metabolize glucose and oxygen, and energy is produced, we see a color change in the mitochondria. And that color change that you can see glowing there is due to a specific enzyme called cytochrome C oxidase. So, the video's, there you go. So every time that cytochrome C oxidase passes an electron to oxygen right at the end of metabolism, it changes color. And that is the principle of which these measurements that I make are on. So I'll go into more detail about that. So near-infrared spectroscopy, for those that aren't familiar with it, is the principle of using near-infrared light, which you can see in the picture of the hand here, travels really well through human tissue. We want to use the absorption properties of chromophores inside the tissue to understand measurements in the tissue. So you can see from this schematic on the right, the basic principle of the technique is that we shine light into the brain. Because it's infrared, it travels quite well through the skull. Light within the brain gets absorbed by specific chromophores such as hemoglobin and cytochrome C oxidase, and the light that we can detect back out, we can use the absorption changes to relate to concentration changes. So this diagram here shows you that principle more clearly, and you can take the change in the attenuation and relate it to the change in the concentration. Most commercial NIRS systems only look at two chromophores within the tissue, which are oxy and deoxyhemoglobin. And you can see in the near-infrared range, they have very different absorption spectrum. So by using a couple of wavelengths, we're able to quantify the concentration changes within that tissue. And so this gives us a way of measuring vascular oxygenation and hemoglobin changes non-invasively, non-ionizingly, in a method that's very portable and safe, and inexpensive as well. Some of the disadvantages of NIRS are that there are actually not just oxy and deoxyhemoglobin in the tissue, there's other chromophores going on. So we have to make lots of assumptions about what's going on underneath the tissue. Also, the tissue that's of interest is sometimes underneath other tissues, such as the skull and scalp, which makes the measurement a bit challenging, and that's why there's a lot of work on the optics of the measurement. So whereas most commercial systems are up here on this diagram, measuring changes in vascular oxygenation via oxy and deoxyhemoglobin, what my group in real London at UCL have been focusing on is measuring changes right down into the cell with cytochrome C oxidase. So this here shows the absorption spectra of oxy and deoxyhemoglobin, and if we use a couple of wavelengths either side of the isobestic point, we're able to resolve concentration changes in them quite accurately. But we're more interested in, well, equally interested in measuring cytochrome C oxidase, so looking at the metabolism of oxygen. And the absorption spectrum of cytochrome C oxidase has this broad peak around 840 nanometers, and that is dependent on the oxidation state of that enzyme. So every time there is as you saw in the video, when electrons are passing through cytochrome C oxidase, we see an absorption change. So using broadband near-infrared spectroscopy, so the whole spectrum across this broad peak around 840 nanometers, we're able to monitor the metabolism of oxygen at a cellular level. So that's what I've been working on And my PhD, in fact, was on building a system I named cerebral, for use broadband near-infrared spectroscopy in the intensive care unit. So broadband near-infrared spectroscopy is very similar to conventional near-infrared spectroscopies, but you need a broadband light source. So in our case, we're using a very simple tungsten halogen bulb. We illuminate tissue via some fiber optics that we place on the head of the baby. And the light is collected by another set of fiber optics, and that goes to a spectrometer. So using the spectrometer in a very high sensitivity CCD, we're able to record the spectral changes of the tissue due to changes in cytochrome C oxidase as well as hemoglobin. So if you're interested more about the system, we published this work a few years ago. This here shows a picture of the system in the intensive care unit. So as you can see, the infant is here in the cot behind the chalkboard, and then there's lots and lots of other systemic monitors, including EEG here as well. And then we have got our infrared system here. So there is a light source here. The black lines are the optical fibers which take light to the baby's forehead, and this is our spectrometer. So we've been demonstrating the use of the system in a challenging environment such as the NICU. So I'll just tell you a bit about hypoxic ischemic encephalopathy. So that's our primary target population of interest. It's relatively common. It occurs in 1 or 2 births per 1,000. It's the largest cause of cerebral palsy in children. And currently it's monitored continuously from birth using EEG. And the gold standard marker for predicting the outcome of the injury is magnetic resonance spectroscopy measurements of the metabolites. So looking at the hydrogen magnetic resonance spectroscopy, we're able to look at the changes in the lactate and the NAA. So the NAA is a marker of neuronal function and health. And looking at this ratio, you can see that in more severe injury, there will be an increased lactate and a reduced NAA compared to normal injury, or normal healthy tissue. The problem is that this very sensitive biomarker can only be performed towards the end of the treatment. So these babies are treated using therapeutic hypothermia, so the babies are cooled down in intensive care to 33.5 degrees C. And during that period, over the first 3 days of life, they aren't able to have this MR scan. So what is really needed in the clinic is a continuous measure of cerebral metabolism that we are able to apply at the bedside. And that's where our near-infrared spectroscopy comes in. There are many different ways of measuring metabolism. You can use a combination of optical techniques, DCS and near-infrared spectroscopy, to measure the cerebral metabolic rates of oxygen. Or you can use a combination of MR, ASL to measure blood flow, and NIRS, again, to get CMRO2. Where we've been focusing on so far is measuring cytochrome C oxidase with broadband NIRS, as it's a slightly simpler equipment that just needs a single machine. I'll just take you through the typical time course of hypoxic ischemic encephalopathy. So the baby is born typically at term, and there'll be a complication during birth. During the injury, the cerebral function and metabolism will be decreased. When the baby's born, it will be resuscitated, and there's normally a period of normal cerebral function, but that can be preceded by a decrease in metabolic function, and that can cause further injury. So this is known as secondary energy failure. The current treatment time is in the first 3 days using therapeutic hypothermia to try and protect brain function. And this is the point where we're interested in trying to monitor cerebral metabolism so that we will be able to guide treatments and give clinicians an idea of what the cerebral metabolic function is like in this period. So the aims of this work is first to show that using broadband NIRS is feasible in the neonatal intensive care unit from the very first day of life. Then we wanted to investigate the relationship between metabolism and oxygenation. And our ultimate goal is to try and find a metabolic biomarker of injury severity that we can give to clinicians at a much earlier stage than they can get to the MR. So this figure up here shows a typical timeline of one of our experiments. So the baby will be born and immediately started cooling treatment for the first 72 hours. The baby is continually monitored with systemic monitoring and EEG as is clinical practice. And then we apply our broadband NIRS machine to the forehead every day from the first day of life for 2 or 3 hours to try and get an understanding of the metabolic function and how it progresses with age. We also apply it during the rewarming period, so the baby is brought out of therapeutic hypothermia slowly, so we're interested in how our metabolism is functioning during the rewarming, and we also record NIRS data on the same day as the MRS scan so that we can compare it to the gold standard. This diagram here shows all of the systemic monitors, so we're measuring blood pressure, arterial saturation, heart rate, respiration rate. We also have EEG, and then this is our broadband NIRS over the frontal hemisphere. And this image here shows a baby with the probes on the head. So this black probe here is 3D printed optode holders, so they're holding the optical fibers onto the head. And we've performed this measurement in over 70 babies now, and we've had success with fitting our instrument into the day-to-day care in the NICU. So the first analysis I'm going to show you is looking at pressure passivity of the metabolism. This study was done in 23 of the babies studied, and they were all around 2 days old at the time of study. They're all term and have healthy birth weights, and they all experienced hypoxic ischemic injury at birth. So the idea behind this study was to use wavelet analysis to identify coherence and semblance between oxygenation and metabolism to quantify a metabolic reactivity. So what that means is looking at the time course of the signals that we record, compare them to the systemic signals to try and understand the relationship between those and whether that is related to injury severity. So this figure shows a data set from one of the more mild injured babies that we've recorded. So the top red line here is the arterial blood pressure, and this fluctuates over an hour of time. And this here is our cytochrome C oxidase measurement, so our metabolic measurement. And you can also see some similar fluctuations. These two plots here show the wavelet analysis. So we're looking here at the coherence, so that's how much the signals vary with each other. And that's on a scale from 0 to 1. So the darker patches show the higher coherence between the two signals, and this is in frequency domain. The semblance here is actually the phase difference between the two. So this is how one signal may be affecting the other, so looking at the phase changes. And the red is more severe, more higher phase relationship. And this baby, which is determined to be quite a mild injury, you can see there's no really overall strong color coming through. Whereas if I compare it to a baby that has a severe injury, so the lactate NAA shows a more severe injury, you can see there's a much more red, strong semblance between the signals. And you can even visually see that. So when there is a large change in the blood pressure, you see a delay, but a similar change in the metabolism. And so this is identified as a severe injury, and this is representative of our study. We found that, in general, if we just look at this graph here to start with, the babies that are more injured, so the more injured is up here on the y-axis, have a stronger semblance between the metabolism and blood pressure. So we're calling this a metabolic reactivity to blood pressure, and the babies with higher injury have a higher dependence of the metabolism on blood pressure. We also looked at the the group of these babies, so if you look at all the babies that were determined to be mild by the lactate NAA, you see a much lower metabolic semblance than the babies that were more severe. But this wasn't seen in any of the other signals, so the relationship between oxygenation and blood pressure, which you might expect as a cerebral autoregulation effect, wasn't seen. And further, we also looked at the outcome. So after 18 months, the babies were assessed using a Bailey test to assess their neurodevelopmental function. And these results were seen as well. So babies that died or have a poor motor outcome had a much stronger semblance or metabolic reactivity than babies who have a good outcome. So these results show that on the second day of life, we're able to look at the difference between the phase between the metabolism and the blood pressure and predict neurodevelopmental outcome at 18 months. So this is a summary of our results. So hopefully we're going to continue to expand our analysis. We have almost 70 babies studied now, so we'll expand that to depth. And hopefully this can become a marker of cerebral health in the first days of life. So moving on to my next analysis. Again, we wanted to investigate the relationship between metabolism and oxygenation. But because we're measuring differential signals, so we're looking at the change in metabolism and oxygenation from an arbitrary baseline, we used spontaneous events. So these babies in intensive care have spontaneous desaturation events. And we use them as a challenge of the oxygenation metabolism to see if we can understand it. So in this study, we were also looking at the same set of babies, 24 this time, and the period of study we're looking at was during the hypothermic treatment, so over the first 4 days of life. So the aim was to examine the relationship between oxygenation and metabolism during these spontaneous events, and our hypothesis was that the relationship would be dependent on the metabolic function, and that function would be related to injury. So this is an example of pulse oximeter data of arterial saturation. So these are two typical events that may occur. So the baby is happily at 95 or 96% saturation, and then suddenly has this desaturation down to 80%, which is a very low level of hypoxia. Same with this baby, we're dropping down to 75. So this is a spontaneous occurring event. This here shows our NIRS signals recorded during that time, so the grey boxes during the desaturation. So you can see a decrease in oxyhemoglobin in red in both babies, and a slight increase in deoxyhemoglobin, which is expected. The difference here is in the cytochrome signal, which is in green. You can see that during this event, the oxygen, the metabolism also drops during the desaturation, whereas that wasn't seen at all in this baby. And if we look at the relationship between the cytochrome and the hemoglobin difference, you can see that during this period there is a strong relationship between cytochrome and oxygenation that wasn't seen in this baby. And after looking at the MRS results, we determined that this was a severe injury and this is a mild injury. So from what we see here in these two case studies, we actually saw it on a group level. So this graph shows the change in oxygenation during desaturation. The black group is the severe injury, and the lighter group is the more mild injury. And you can see that the oxygenation drop during desaturation is very similar. The hemoglobin total, so blood volume, increased in both groups. But the biggest difference here is that the cytochrome signal in the mild group stayed very stable during desaturation, whereas in the severe group you see a significant drop in metabolism. This is also seen if you look at the cerebral oxygenation against cytochrome, so a very stable change in the metabolism in the brain, even though there is a large oxygenation drop for the mild group, but a drop in cytochrome for the severe group. And if we looked at the gradients on an individual level, we found that the gradient of the oxygenation and metabolism was predictive of injury. So a very strong relationship, a decrease in metabolism with oxygenation predicts more severe injury. Thinking about the mechanism behind this, in the literature there's a lot of biphasic changes in metabolism with blood flow. So we expect that in this mild group, while the tissue oxygenation decreases, even though there's no change in metabolism, at a certain point, a threshold, you will see the reduction in metabolism. Whereas the severe group, they're already at that threshold, so you see this immediate decrease. And we believe that this is related to the metabolic rate, which is related to the metabolic function and the injury. So to conclude this analysis, we've shown that even though there is a similar oxygenation change during desaturation, we see differences in the mitochondrial changes that is dependent on injury severity. We think that's to do to the differences in cerebral metabolism, and that's been shown by other groups before us. And again, this is showing that we have another early, real-time, continuous marker of cerebral metabolic function in HIE. So finally, I want to show you a case study that where we saw seizures in one of our HIE infants. This baby was having recurrent seizures, so we saw 5 seizures continuously. The baby was termed but was delivered by emergency cesarean section and was receiving therapeutic hypothermia up until day 4. And then during rewarming, the baby was developed these seizures. So this shows the EEG and the amplitude integrated EEG showing the 5 recurrent seizures, and these were identified by a neonatologist. And during the seizure period, we were able to apply our broadband NIRS to monitor cerebral hemodynamics and metabolism. So this figure here shows in purple the amplitude integrated EEG, and the lines here show the start and end of each seizure period as determined by a neonatologist. And the black and red line here show the hemodynamic measurements that we recorded during seizures with broadband NIRS, and the green shows the cytochrome changes. And what's interesting here is that at each seizure we saw a decrease in total hemoglobin, so blood volume, a decrease in cerebral oxygenation, but an increase in cerebral metabolism. The other thing that was interesting is that whereas the hemodynamics returned to a baseline after each seizure, we saw a consistent decrease in the cytochrome signal, so potentially monitoring damage to metabolism here. And further, if we zoom into each of the seizures, you can see that there was a difference in the time course of the hemodynamic traces and the electrical activity from the EEG and the metabolism. So if we look on seizure 2, for example, there's a decrease in oxygenation and total hemoglobin slightly before the seizure was identified, and that continued and increased back to normal, whereas the cytochrome signal increased with the seizure. So we saw a much stronger relationship between the electrical activity and the cerebral metabolism found with blood flow, blood volume and oxygenation. So this is the first time that metabolism has been recorded during seizures, and we're very interested in this fall of the cytochrome baseline during seizures because potentially we may have a monitor of injury on a cellular level. Okay, so that's all of the work that I'm going to present from our studies in Rio London. Here I'm going to just give a brief overview of what we've been doing here in Fake London, and in collaboration with Keith's lab here. So we want to quantify exactly what our cytochrome measurement is recording during HIE. So we've gone back to an animal model. So we're using a piglet model of hypoxic ischemia, and we're monitoring continuously using broadband NIRS before, during an insult, and afterwards. We're performing some oxygen challenges, so similar to the spontaneous desaturation events that we saw in the baby data. We're performing these challenges before and after the insult to see if we're able to identify metabolic changes caused by the insult. And we're doing it in 3 groups, so a control group which doesn't have an insult, a moderate group which has a 10-minute insult, so dropping the oxygenation and clamping the carotid arteries for 10 minutes, and a severe group. And the other thing we were very interested to see is if our cytochrome signal is able to identify the differences between a moderate and severe insult, which would be very useful for clinicians in the NICU. So in collaboration with AJ and Matt, we've developed a broadband NIRS and DCS system. So DCS is diffuse correlation spectroscopy, and that's able to measure an index of blood flow. Using a system of shutters and optical fibers, we're able to simultaneously measure changes in hemoglobin oxygenation, cytochrome C oxidase, and blood flow. And this is the first time this has been done. So there's 4 fibers that are held onto the piglet's head, and we're continuously recording with a shutter system. So this graph here shows, an example of some of the data that we've been recording during insult. So here is a baseline period, and then if you follow the black line, you're looking at the DCS recording of blood flow during the insult. So you see a massive decrease in blood flow, and then a recovery when we release the insult. The red and the blue lines show oxy and deoxyhemoglobin, so you see a very similar trend with the oxy and deoxyhemoglobin as they decrease during the insult. And then the green line, which is a bit faint, I'm sorry, but is the cytochrome C oxidase signal. So you can see a marked difference between, firstly, when the metabolism starts to decrease, but also the fact that even though the blood flow and the oxygenation has plateaued, we still see a consistent decrease in metabolism, which is possibly representing the continued depletion of cellular energy. This graph here shows a severe injury, so this is a 10-minute insult, and we see the same changes, so a plateau of the blood flow and the oxygenation, and a consistent decrease even for up to 20 minutes in metabolism. So this is very new and exciting data. We've performed over 30 piglet studies now, and we're going to be analyzing this data and also combining it with other measures of metabolism, such as FDG PET to measure glucose metabolism, MRS to measure the lactate, and we can combine the blood flow and the hemodynamics to get CMRO2. So this is going to be a very nice cohesive study of lots of different measures of metabolism to try and understand what is the best metabolic marker. And perhaps it's a combination of all these techniques to understand the severity of injury. So our future work will be hoping to get broadband NIRS and DCS combined into the NICU to get real-time measurements of oxygenation metabolism. So in conclusion, the work I presented here has shown that firstly, optical measurement is feasible in the NICU. We're seeing that there is a strong relationship between metabolism and oxygenation, indicating severe injury, and also a strong relationship between metabolism and blood pressure, also indicates severe injury. I showed you some seizure analysis, and I think the overall message is that optical measurements and metabolism is really important in the NICU, and hopefully with developments in both the optics and understanding of metabolism, we can see this in the clinic sometime soon. So I'd like to thank the team back at UCL. We're a very diverse team of clinicians, physicists and engineers, and the team here in London. I've really enjoyed my time here, and I'd like to thank everyone involved. Thank you very much.
Questions for Gemma?
And that was a very nice presentation. Some of the best explanation of cytochrome oxidase I've seen. The HIE group, presumably have some blood pressure correlations throughout that duration of measurement.
Blood pressure correlations?
During the phases that you were measuring, do you have any perfusion data or blood pressure?
Yeah, so the Wavelet analysis, we look at the blood pressure data to try and, so we were using the oscillations in the blood pressure with the oscillations in metabolism and oxygenation to try and look at the relationship. Because our first hypothesis would be that the strength of relationship between blood pressure and oxygenation in the brain would indicate a failure of cerebral auto-regulation, especially in the more severe infants. We didn't see that. What we saw was the correlation between blood pressure and metabolism was indicative of injury.
And I see the placement of the probes around the forehead.
Yeah, so that is more of a practicality than a decision that we made. So the infants all have full head EEG. That's a clinical decision. So the easiest place to access is the forehead. Also, we avoid problems with hair on the forehead. We are developing a new system that will have multiple channels, so we'll be able to cover the whole head. But I don't think we'll be able to do continuous monitoring with that. It might be more of a, one measurement per day situation, just because of the practicalities in the NICU.
I was actually really interested in your correlation data. So you thought that it was the severe injuries that reduced metabolism?
Yeah.
So I was wondering if it's more likely that the blood flow is actually growing acid, and that's making it less tolerant
Yeah.
to fluctuations.
Yeah, so that we would love to have the measurement of blood flow in the NICU, which is what we're working towards. That's a really good hypothesis. I think, thinking about the relationship between blood flow and metabolism, so I think the change that you've reported, I think that, it might be that we're observing the effect of a reduced blood flow. That's why we're seeing people raising.
Have you ever tried to correlate the change in the oxygenation signal and the HT signal that's supposed to be related to blood flow to the change in cycles? Is there any relationship between those?
So that's what the correlation was.
Shows that actually. You don't see a correlation.
So this is
severe injury. If you go back.
so this is HT difference bedside. So HT difference is supposed to be related to blood flow, but that's only during when arterial saturation is stable. So here our challenge is a decrease in saturation. So I don't know whether that uncouples from blood flow then. But it would be great to have blood flow during this period.
Definitely. Is there last chance before she heads off to the real one? Yeah.
Your seizure data that you had, do you know what the seizure was? Was it an optical lobe, clinical lobe, or?
That's the seizure was actually focalized, I think, temporal. So we've actually been recording a few more seizures, and we're working on that data now. And it's very interesting to see, firstly, the difference between the electrical and activity in the blood, the hemoglobins, and whether that's because of a delay because we're recording frontally rather than
In one subject, it almost looks like there's a spike in your cytochrome before the seizure happens.
Yeah, so
and it just was that one, I think it's that one seizure, so that's pretty
Yeah. So we're thinking maybe that cytochrome might be a good way of detecting seizures because you see the EEG signals and it's pretty difficult to pull out the seizures. Whereas with the cytochrome, perhaps in combination with EEG, we might have a seizure marker there.
Right, so this is going to be the key question. So I remember sitting in class my next level, we would talk about having problems with NIRS in adults because it's called a thicker
Yeah.
compared to immunity. So if we are to say try this in an adult skull, like an adult head, what kind of problems do you have trying to get the signal to the skull?
So NIRS has been done a lot in adults. You can vary the source and detector distance to make sure that you're obtaining data from the cortex. Yeah, I think that's definitely possible.
Thanks.
I have a question. I'm just trying to get a handle on your set-frequency oxidase
Yes.
traces and what they actually mean. So based on your video, you showed this very nice video where you showed that the change in color is representative of
The oxidation
the
state.
oxidation.
Yeah.
So I would assume in that situation that the amount of set-frequency oxidase that you have in oxidized versus reduced form is representative of those rates. Is that correct?
Yes.
And then for hemoglobin, it seems to me that it's kind of, it's not the rate, it's just how much is there.
Yeah, so the thing with hemoglobin is that you can measure oxygen when it's bound to hemoglobin, but it doesn't give you an idea of, firstly, whether there's oxygen in the blood or in the plasma, whether there's oxygen being delivered to the cells, and whether there's uptake by the cells and actually metabolism occurring. So it is nice so that we have a cellular monitoring of the oxygen consumption.
I'm just trying.
Oh, so in terms of what the signal means,
Your signals, does the green curve maybe represent more of a rate or a derivative versus the black and red curve?
yeah, so it's,
Concentration.
so we're actually looking at the concentration of oxidized cytochrome C oxidase. So, and metabolism becomes oxygen limited. If metabolism becomes oxygen limited, then we'll see a decrease in oxidized cytochrome. So, yeah, for example, in this figure, the increase in cytochrome or oxidized cytochrome is representative of an increase in oxygen uptake, so an increase in oxidative metabolism. Does that make sense?
Yeah.
Okay.